So, welcome you all for the fourth edition of Echo Resonance. The Department of Respiratory Medicine is eager to continue this learning initiative. We started almost 9 to 10 months back. As you all know, Echo is an extension for community healthcare outcomes. So this is the fourth session, and this session will focus on understanding the COPD phenotypes and the ongoing state of our research in the field. So to enrich the session, Dr. Angira Das Gupta Madam and Dr. Pongali B. Raghavendra Sir, both of them have consented to be part of the program, and we are eager to learn from them on the cutting edge technology in the field of COPD. First, Dr. Pongali B. Raghavendra Sir, a brief introduction about him. He's a faculty and senior research scientist of human disease biology and translational sciences, and he has worked in multiple research facilities across the world, from Michigan State University, USA, to Mahe, Mangalore, and from earning a PhD degree from the Center for DNA Fingerprinting and Diagnostics, Hyderabad in 2010. He has multiple innovations to his credit, starting from his basic degree in 2002 and to his PhD. And he was the best and first prize winner from the Science, Technology and Product Association Award in the year 2015. He has got grants worth more than 2 crores, and he is currently working on multiple research products, areas across the field of biotechnology, and in particular, for our interest, it is airway diseases, and especially COPD. And he has recently started a research with respect to COPD, the short name FAST COPD, factors affecting survival in severe and very severe COPD after admission in ICUs of tertiary care centers of India, which multiple centers across India are part of that. Respected sir, welcome you to the program, and the stage is yours now.
Yeah, thank you, Dr. I would first thank Dr. Palnivel and then the team members for inviting me for this session. I would like to share the screen so that we can. Is the screen visible to everyone?
Yes, sir. Due to some technical issues, we had started the program a little late. From the organizing team, our sincere apologies for the technical difficulties caused. So to make up for the time, so we'll be having a approximately 30-minute session by both the two eminent faculties, so that we can wind up on time.
Yeah. So can I start now then?
Sure, sir. Thank you, sir.
Yeah, so in broad, as I was saying, that we are working on the biology of chronic and infectious disease models by and large. So I represent National Institute for Biomedical Genomics. It's a DBT institute. So for today's talk, I thought to first just in a brief, because I was expecting that my turn was second, but looks like it was first now. But still, you've seen on a screen couple of numbers with gadgets with some numbers. I'm sure you will be wondering that this is a COPD guy, a COPD talk, and this guy is showing these kind of things. But still, the broader question of phenotype, I thought that Dr. Angira would cover how and what. But why do we need to really focus on why is also a very important question. That's why I think the number one stage always goes with a branded company, where they start to ask the question why, rather than first looking at how and what the events are occurring. To start with this, and again in very broader context, because I know that there are multiple medical fraternity people sitting there. You know that in Indian things, it is challenging to be a clinical scientist, because you know that it is a very long period journey, which I think most of the people are kind of not so much keen in going down. So and these sessions, and I think your HOD and others are also trying to make sure that to see that which path has to be taken. Whether this, I know that whether it will be a little very intruding questions to you, whether you have to go on your old way of path, which the people have been doing it, or taking again a new path. So again, I think by the end of this talk, I think probably if we can ignite some of them to see that what could be best way to choose an path also could be an very interesting way of taking forward. But anyway, I have quoted the journal which has been published in Indian Institute of Science, which says that which could identify and suggest possible solutions for challenges in Indian clinical scientists. Okay, let's come to our basic talk for today. What we were discussing was like translating the omics experimental approaches in pulmonary disorders and to potential clinical applications. So by and large, our lab has been doing from past 8 to 10 years in this area of looking at COPD, bronchitis, and overlap of asthmatic cases also, and again looking at the cardiopulmonary and pulmonary and the CVD biology also. So just to brief very quickly, and I don't want to go into the details, but you know very well that COPD and then the kind of most of the things can be taken into and based upon the gold standard classifications, which you say that whether the patient is under a mild, moderate, or a severe condition depending upon FEV1 and FEVC. And then there are multiple factors. We do agree that the smoking and biomass, smoking and the other environmental hazards could be the potential things. And again, biomass also is coming up in and by and large because of the exposure. So having said that, these causes obstruction in this airways to accumulations, and then how these inflammations and infections, basically our lab also looks at the inflammasomes, which causes and the progressive and irreversible decline in the lung functions. Again, incidences, we all know that it is a very highly need of the hour to be concentrated, because in India also many of the states also have this mortality rate, which we have been shown here. And then it's a leading cause. And no doubt that much further research has to be implemented in these areas. So today, we would like to just again brief some biological questions, biomedical questions, what we try to address and what we try to look at the phenotype or progression and the regulation applications. Why does this individual susceptibility occurs? Why do some patients get it, or why do some individuals get prone into this kind of diseases, whether it is a genetic or environmental triggers, or why there is an heterogeneity or a manifestation in this progression mode, why some people are mild, while moderate, and some people are extremely severe. So what is the classification of these cause, and then what could be the pathology and function? We try to look at the systemic effects and the immunomodulations, and whether comorbidities also, like a couple of diabetes or hypertension affects the COPD much more. And then finally, not but the least, again, trying to very keenly address with some stem cells and peptide biologies, which we have been generating in our lab, to address some translational or looking at the disease prognosis. So by giving this broader picture, today I will be focusing on giving some omics-based studies, which we have been using with the clinical applications or clinical samples rather than individuals. And then how do it really helps us to look into the translational biology. So when I say this, as I said that our lab has been working on, like couple of pyrethroids, which are affecting the lungs, and then looking at the SARS-CoV based variants and different variants, how it impacts not only SARS and MERS also, what could be the receptor levels, and what other different conditions also we have been looking in our things, and all our other things are in progress. Again, cardiac biology also, we try to look at the overall impact of cardiopulmonary things in the phenotypes of long COVID, and then again look at the immunogenetic determinants, why younger, and we have a pool of cohort from South India where only the young patients are getting into that, and the cause and causality has not been described in an Indian context. So we are trying to get to look at this sequelae, cardiovascular pulmonary sequelae after the long COVID impacts. Again, as I said that we have some translational biology concepts where we develop stem cells and then the peptides also, so that we treat the things. And we have been successfully able to keep an human clinical cohort with the pulmonary samples, both sputum, blood, and kind of cell lines, and then cardiac, and then the other related stem cell samples. To just go to a brief, these are the gene variants associated overall in the COPD in the Indian subpopulations. If you see that, majorly the MMPs, metallomatrix proteins, or GST, where basically the glutathione transferase, because redox plays a key role, and again IREB, where you see the iron responsive elements. And why this disparity between as a whole when we have an Indian population? The question immediately arises that why the influence of environmental trigger would be a question, but still how this gene gets into a pathogenic allele when it could be a same disease from South India, North India, or central part of India, or any other thing. So we have described here, and we have been tracking out with mining, with seeing that what are the major impacting gene variants in the Indian subpopulation. So having said that, I am sure that most of you people, though the layman also was very much aware from 2020 to 2021 that everyone starts to say RT-PCR, RT-PCR. So what it goes is like because it was a major thing with COVID-19, and they had to make these reports and make sure that you get a negative report only, then you are allowed to travel or move it freely. So as you know that it is a very simplistic thing, like a polymerase chain reaction, we call it as PCR, and then it is tagged with an fluorescent like kind of a dye, cyber green or anything. You do a denaturations at high temperature, annealing and elongations, and then measure the amplification rate. And basically, depending upon what you want to see, it could be the gene what you want. Here it could be like a viral component you want to see. So it goes into like an to explain very briefly, you have a template DNA, and then first cycle, second cycle, like that multiple copies are generated, and then overall there will be an exponential amplifications. So you have the very much of very much of a key word called as CT value, which was lingering around for a long years. So it is called as cycle threshold. So CT values are important and very significant to detect the reactions, detection of the amplification, and it is a parameter used for the quantification. If you have a higher CT, that means that there is no amplification. When you have a lower CT values, that means you have a higher amplification, which is briefly explained in this technique. And absolute quantification, relative quantification, comparative quantification. So these can be measured in the PCR when you are applying for any kind of methodologies. And as you know that it is a very kind of an basic experiment, but still it explains some sensitivity, specificity, a low template, rapid procedure to understand the diagnosis, or again genotyping, microRNA. All these I have mentioned that what all could be a beneficial. So this could be a basic application. Now moving on to a little gears, what I said that I wanted to give a feel that what can be done with a nucleic acid that is called as a DNA. Now, what specific project we are working is like kind of identifying these gene variants or a microbial dynamics in this COPD samples versus control samples in the eastern and versus southern, and its association with the disease severity. Again, it is very clear from this paper which has been published in our institute only. It is called Genomatia, and we have also made a portal which is available freely by DBT. If you see that there is a heat map difference in allele frequencies for RSIDs between populations of east and south demography. For example, there can be an Dravidian population, or there can be a different population of between the Indian within the Indian things are east and south also, where the average internal populations within this region is also taken, and you get an RSIDs which will be specific and we are looking for specifically the COPD associated things across the Indian geographical locations. When we say that, it is a very simplistic again to understanding. We are bringing the workflow. We also, I have been associated with you people from past 4 years. MGMR CRI also sends us the samples from Pondicherry, from Ramaya, Bangalore, and JSS Mysore. We have been using, and eastern part we have been doing with AIIMS Kalyani and BRC hospitals from eastern part. So what we collect is we collect the whole blood, and then we collect the sputum, and then induced sputum, when I say sputum, and then we do a DNA isolation and do an whole exome sequencings, serum separations for proteomics study and ELISAs. Again, immune linked, enzyme linked immunosorbital assays to know the antigen antibody relation, and then PBMC isolations. It's a primary cell line to address the PCRs and Western blot methodologies for functional studies. So these all explains us what is the association between the disease heterogeneity. And finally, again, as I said that sputum also, we collect a DNA from sputum, and then we do a shotgun based 16S RNA sequencing, where I'll be showing a couple of results. So overall, I wanted to say that with a biological sample, what all could be done in by and large by using the biological material. So when we collect these samples, each drop of the blood is important for us, and each material of the content of sputum is also maximum utilized at the framework of the, again, following the IRB rules, and then again using the quantification of as much as results, so that you can associate the disease heterogeneity. When I say that, again, this is just again a brief clinical metadata which you people are very much aware, and I did not explain, but just making sure that we also include the inclusion and exclusion criteria with our framework, and then we include the smokers and non-smokers and biomass, and then this is what we have here. If you see the percentages of 39, 36 and 25% among the smokers to non-smokers in the southern populations, and you have 58% smokers in the COPD in eastern things compared with 17 to 25%. So this I am talking about the cohort with which I am giving a brief example of 100 samples which we have collected, and we have been doing the process, but the data has been generated. So again, it is a very clear indication. You have a gold 1 standard 9 and gold 250 and gold 310. As I said, that gold 2 would be more because the people going into the moderate to severe will be as age progresses, and then it depends upon the mutations and gene alleles what we have seen. And this is to giving an example of when we do a 16S RNA, how do you find? So you can clearly see that what is the difference between the phylum or between the southern and then eastern thing. So your actinobacteria, firmicide, phosphobacteria, and many more to be quantified, and their relative abundance can be really made sure with the p-value, which is we use an Wilcoxon rank sum test. And this is a brief idea to show you that what could be the percentage of microbial species present in the samples. Same way here we are showing the genus, where you say that between the differences between the south and east, again it gives you the phylum, it gives you genus, and then you have to really quantify. If you see that in the circle you have a different kinds of colors which are represented for actinomycetes, Bifidobacterium, Fusobacterium, and other things. So this is just an example to show that in a 100 population, as you increase the number, you can clearly see a demarcation between the southern and the eastern regions. Again, as I said that we also do proteomics. Again, proteomics is really important to understand that what is the translational machinery, where you have and everyone would have know the central dogma from DNA to RNA and RNA to a protein. So where you are having a mutation in the DNA, then you have a defective protein, or you have a not complete formation of the protein, or you get the the kind of we evaluate plasma-based proteomic analysis from the healthy and COPD patients, and again here I am showing you the results of the southern or to the south patients, or to my left hand side, and eastern patients are to your right hand side. If when we do this substantially also, we see a clear heterogeneity data set between observation between the two population based. So this is a pilot study, and then when you increase the numbers, you get a differential heterogenic pool of frequency. To your right hand side, you can see the rho max, which is from red color to rho minimum is blue, and rho max is going with the red color, where you have a different implication. If you see S1, S4, S6, S7, which is been represented here, you can clearly see that there is a differential kind of protein network when compared to the others. And when you see the E1, E2, E4, E7, E9, you can it also implies very clearly. So overall, this we have generated some top hit of proteins which are being getting into differential zone, which we are not interested in the 99% expressed one. The pool which is rarely expressed, right? It could be 1% or a 2%. We concentrate on that proteins because they will be your key players to judge between what is the difference between your southern and eastern things. So this is again one more example of omics, which you have seen for the microbiome, which we do as an kind of an microbiome based omics, and then this is an example for showing for a plasma proteomics. Again, this is done by LCMS method, and these all pipelines have been developed in the institute, and our institute is a cutting edge technologies we use for doing this thing. So overall, to say that we want to identify the protein signatures between the healthy and the COPD patients, between southern and the eastern samples. So moving little bit gears to little bit to an next level. So okay, I have done a PCR, and still I feel that I have a large number, but the data which I am mining is in very much limited, because when you do a PCR, you have to dedicate yourself. Probably you can do maximum in a 96 well plate with an one kind of 5 primers or 10 primers, and you can only identify 10 genes or 20 genes. So if I want to push my accelerator more and want to go into much more details, then it would be like whole exome sequencing is the best bet what we do. You have again a sample from whatever source. It could be like kind of a sputum, or it could be from the blood. You isolate a DNA, then you make a library preparation based upon the exons, and then you make an exome enrichment, and then you make an amplification, and then you do a sequencing. So we have an Illumina sequencer and next generation sequencers in our institute, where again data analysis is done at a higher level. Then you have a chance of looking at thousands of genes and what is happening. And then we also have a network established with our bioinformatic team, who do us with a sequencing. We get a raw data, which we call it as a SEF file, and whatever you get a sequencing, then you use a FastQ, SAM and BAM tools are being used, and finally you generate an VCF files, and then that will explain you that what are the defective genes which are present in your sample, and by and large. So this is just again giving you a pipeline where whole exome sequencing is being done in Indian COPD patients, distribution among southern and eastern. So you have an raw sequencing data FastQ format. Quality control is made so that you remove the low quality bases. That's called as, in other words, called as adapter trimming. So you use FastQ and Trimomatic tools. Then you pre-process, align the human genome sequencing, duplicating is removed. Then base quality recalibration is done, QC alignment data is done. You use WAMEM and GATA4 tools for this. Then germline variant calling is done, stringent filtering of artifact variants is done, then annotations have been done, and potentially the rare variant identification is done. So this description is very clear to showing you the processing and analysis which we do. And here we have a data for showing between the eastern and southern samples. So we want to identify the gene signatures by doing whole exome sequencings. So distribution of average of 50 samples is being shown here after alignment. If you see that it includes some controls also, and then it includes from sample IDs of southern and then eastern samples. So the pilot data says that the average coverage was 89x range, 68 to 103x. Median coverage was for your 90x. And this is the distribution coverage what I am talking about, and this also implies. So then we found top 10 frequently mutated genes at the germline mutations which have potentially damaging effect. If you see that again, we have given you at the top end the biomass exposure, who are all yes and who are all not applicable and who are all no. The smoking history of the patients. So this compartment is for your eastern samples, though I agree that there is a little smaller number, but we are already in the process of increasing our sample number because statistically it should be more relevant. Then this is the southern populations. If you see that these are the top 10 missense mutations which is been represented with the data, and this is the frequency 15, 13, 10% and what. So this gives us very, I know that it is a very one-page graph, but this is a huge work where you need to do an analysis of common variants and case controlling between the controls and the COPD patients. And these all are the couple of the genes which we found, and these are important as a calcium regulators or an iron regulators or other things. So still we are under the process. So what I want to convey is that we have done microbiomics, we have done plasma proteomics, and then we are doing it and then for the gene signatures by doing the whole exome sequencing. So these are the major methodologies which we do. And to brief, we as I'm saying that this could be to show represent that healthy and COPD patients description, and it is like an overlap of an cardiopulmonary also. And overall, we want to look at the metabolomics. What are the metabolites? We want to look at the genomics. What are the gene level? Transcriptomics to look at the transcriptional factors, and protein levels by using the proteomics and microbiomes. So you associate everything, and then you from a patient group and a cohort, then you try to look at a precision medicine, which is individually called as individual tailored precision therapy. So these all will be an research based questions which we do it, and then we get an evidence based factors which we then suggest for a better prognosis with a lesser side effects. And again, I have not shown here the treatment regime, which because you people are more efficient, and which you people are doing on the daily basis to treat it. But again, believe me, there are some kind of caveats where the precision medicine could be concentrated, because you cannot tab, as each one can be individual gene or a protein will be difference among patients to patients. So overall, again, to move, that was like kind of techniques or technologies or applications, what omics, which we have been calling. And this we have been doing in Bangalore when I initially started in 2013. So this is a bioaerosol method. So when you breathe, because Bangalore city has a very much of highly pollen thing, and then when you see the OPD cases, most of the winter season and the monsoon season, you get the patients with highly in the OPD with respiratory disorders. These are mostly with again leading to asthmatic and then the COPD conditions. And then this is a brief area where, I don't know, you may be aware of little bit of Bangalore geography. This is a central silk board area where we found that there is an average of high range of pollution and the pollutants. This corresponding data has been taken from the geographical department of Karnataka, but still, what is the ambient air standards, which we have been talking a lot in Indian things? What is the particulate matter? What is the size of the particulate matter, and how good these can be an influential for the thing. So we have developed these aerosol models also, where we collect the sample and then we do a 16S RNA, then we do it for biochemical assays, looking for different things, like by again using the blood and different primary cells, and then we validated, and then we have done some patents also in this for collection of the samples, and then we have found some novel bacterial species also. Then we have also seen that these are the brief assays, which functional assays, which will decipher you that what is the monitoring method of bioaerosol, and how these experimental models of bioaerosol could be in the small animals or collected from the human samples can be more useful. This is again giving a brief of an example to show with the pulmonology department of Ramaya Hospital, where you see a CT analysis of post-COVID how there is a spirometry difference among the patients. So this is a very clear indication of fibrosis of the lung, where you have the influence of the COVID infection, where it is giving an impression, or it is giving the value of PFT results, where it is clearly showing there is a fibrosis. So these kind of patients are very important, and then the studies are important because it gives us better understanding and the causative roots. So to just say that these are the ongoing projects, as I said that we are looking at the genetic variants. So we would like to identify different SNPs, single nucleotide point variants, modifiers, morely potential inflammasome mediated, because we feel that these are the major culprits in this cohort. Then identify the bacterial agents and then susceptible resistant factors. Also look at the host immunity parameters. So again, as I said, that this is one more example to show that we have been pursuing that whether autoimmunity, persistent virus, or viral awakening. This is like a kind of an minor inflammation, which is because it has a difference between the population genetic predisposition and how it is impacting the lung inflammation. And we have developed an animal models and then stem cell models also. So this overall, this is a collective effort from Ramaya, ISC, and NIBMG for this. And then we want to look at the clinical and biological factors of what is happening in the pulmonary sequel of a long COVID patients in Indian cohort. So again, in brief, this is again, before ending, nearing to my talk, I am just trying to say that the clinical scientists could be very much important in Indian aspects to be asking for the new novel questions, expecting the research in clinic and besides and beyond, preparing new therapies and then looking for tailored medicines, and then establishing again preclinical models or tissue banks and other things. So these are the basic, again, what I said that where young vulnerable individuals can be really tracked. And then pre-existing comorbidities, again, you people are the more important people to decipher the clinical epidemiological correlations. Again, look at the biomarker signatures or look at the different functional signatures and then look at the prognosis levels. So these are the exciting areas where we feel or the countries of need where apart from clinics, this has to be extended and should be taken into the larger end. So with this, I think I would like to really end my session. And then here is Ms. Debashmita Banerjee, who is very key doctoral student who has been working with me. And thanks to all the team, my NIBMG and the collaborators from Eastern and Southern and overall country, where we have been starting to develop the cohort, and then we have been consistently, and thanks to the grant supporting agencies also for encouraging our work. And this is my email ID, pbr1@nibmg.ac.in. And I would be very happy that I can really contribute in one way or another in again and be encouraging the youngsters also to participate into the research arena, and novel innovative ideas are always welcome, and we will be happy to accommodate in one form or other. I think I am on right dot with 3:30 is the time what we have been given to me, and then I will be happy to take up any questions.
Thank you, sir. That was a wonderful talk on how cutting-edge research will have a significant effect on patient care. And we are more than interested in knowing and learning more things and more tools so that we can improve the quality of life of our ever-suffering COPD patients. Thank you, sir. Welcome, ma'am. Angela Dasgupta, ma'am. Welcome to the program.
Yes, sir. Thank you. Thank you, Dr. Vimal. Dr. Raghavendra, I'm back. Actually, I just missed your wonderful talk.
No, that's fine. That's fine.
Yeah. So as usual, the last 5 minutes listening to you, it was very much encouraging, even for the youngsters to take up research in COPD. We look forward to further collaborations with you as well. And
I
I'm
think
extremely
someone
sorry
has
for
a question.
Squeezing in of your time.
No, I think someone has a question. I think any role of digital health technology is the question what has been.
Yeah, there is one in the chat.
Yeah, I think I will be very brief in giving you the example. We also use the mobile-based things, and then we are also trying to generate some of the interesting biomarker-based. And we'll be happy to really, because of the constraint of time, I'm not going into the details, but still, yes. to give you answer short that yes, digital health role also technology plays a role, and we can manipulate it in by and large by looking at least to understand the progression and then link up and sending some important key aspects when you are in a rural area to the doctor to understand and help you in the prognosis methodology or a treatment regime.
Yeah, thank you.